Hello, everyone. Welcome to the Department of Psychiatry and Behavioral Sciences Grand Rounds. My name is Joe Ceramelli, and I oversee Grand Rounds for our department. So there's a couple of us that work on Grand Rounds each week, including Samhara Braha and Mike Walker. All of our Grand Rounds presentations are recorded, and we put them on the department website under the Grand Rounds tab. Usually you can access a presentation the following week. During the presentation, if you have any comments or questions, I encourage you to write those into the chat or the Q&A. I'll collect those and go through those at the end with today's presenter. There's two sources of funding for this Grand Round series, the Ripley Fund and the Garvey Institute for Brain Health Solutions. And at the end, close to the end of the presentation, I'll put a link inside the chat that takes you to a short 4 or 5 question evaluation. That's helpful for me in planning Grand Rounds and communicating with speakers. So I'll move on to talking about today's presenter. Today's presenter is Dr. Jeffrey Iliff. And Dr. Iliff is the Associate Director for Research at the Vision 20 Northwest Mental Illness Research Education and Clinical Center, or the MIREC, at the VA Puget Sound. And he's a professor in 2 departments, in psychiatry and behavioral sciences and in neurology here within the UW School of Medicine. And in the Department of Neurology, Dr. Iliff has an endowed position as the Arthur J. and Marcella McCaffrey Professor in Alzheimer's Disease. Just a summary of background, Dr. Iliff had completed undergraduate studies here at the University of Washington, and then a doctorate, postdoctorate, and faculty positions at Oregon Health and Science University. And then in 2019, returned to the University of Washington. Now, Dr. Iliff was part of the research team that discovered what's called the glymphatic system, which is the brain's cleaning system active during sleep. And his research is now focusing on identifying molecular changes underlying the glymphatic system failure that might go along with aging and traumatic brain injury. So with that, I will stop there for now and turn it over to Dr. Iliff.
Okay, can you hear me and see?
Yes.
Excellent. Thank you for that introduction and for the opportunity to speak. This is actually, I believe this is my first psychiatry Grand Rounds since coming to the University of Washington three and a half years ago. So I'm excited for the opportunity to interact with this group. So I'm going to be providing a bit of a whirlwind tour for a bunch of work that has happened over the last 10 years or so, defining what has been termed the glymphatic system. If you don't recognize that word, it's because it's a made-up word. We made it up about 10 years ago when we first described perivascular fluid transport in the CNS, initially in rodents. But I'll be talking today about what we've learned over those 10 years about the function of this system and how a picture is emerging of what its role, or what the role of its dysfunction might be in the development of age-related neurodegenerative processes. So quick disclosures, my lab's funded by the NIH, of course, the Department of Veterans Affairs, Department of Defense, and the UW School of Medicine. I serve as chair for the scientific advisory board for a company called Applied Cognition, and as a consultant to Griffin Biosciences. I do receive compensation and equity stake in both of those companies, and both of those companies are working in the glymphatic space. So that's important to keep in mind as I walk through my presentation. And I'm a full-time federal employee. And the opinions I'm going to provide in the next 45 minutes or so are mine and not those of the federal government. So just a quick view of the lens that my lab brings to the biology that we study. So our lab focuses on neurodegeneration generally and Alzheimer's disease in particular. The things that we're most interested in are the commonalities that link these age-related dementing disorders together. And one of the commonalities that my group tends to focus on is the role that protein misaggregation plays in their pathogenesis. So different neurodegenerative conditions, from Alzheimer's disease to Parkinson's to ALS, they're each characterized by the misaggregation of different peptides and proteins. So in the case of Alzheimer's disease, there's extracellular plaques made up of amyloid beta, and there's intracellular tangles made up of hyperphosphorylated versions of the protein tau. Parkinson's disease and Lewy body dementia are characterized by intracellular alpha-synuclein aggregates. So our question is, what is it that renders the brain vulnerable to this process of protein misaggregation that seems to underlie these different neurodegenerative disorders? By way of clues, our group looks toward non-genetic risk factors as clues into what it is that could be conferring this kind of vulnerability. So we think about things like aging, the presence of some cerebrovascular disease, exposure to traumatic brain injury, including mild traumatic brain injury, and sleep disruption, which have emerged as, in many cases, risk factors that are common across these different conditions. So then the question that our lab focuses on is what changes in the aging or in the injured brain that renders it vulnerable to this process of protein misaggregation or the propagation of aggregates once they're formed. So by way of introduction to the biology that my lab studies, we're going to be talking about cranial fluid dynamics, which is maybe something no one really wants to know about, maybe something that no one's learned much about in their training, but I think it's something we're coming to realize ends up being more important than I think maybe we thought of previously. So cranial fluid dynamics, as we've understood them, really from the dawn of modern, even Enlightenment era medicine up until just a couple of years ago, hasn't undergone a great deal of revision. So what we have understood about cranial fluid dynamics is that the biology and the function of the cerebral spinal fluid, or the CSF, is central to it. So if you learned, most of you learned something about the CSF, at least in medical school, maybe in residency or fellowship. What we do know is that it is secreted by the choroid plexuses. It's within the lateral third and fourth ventricles. It's a relatively protein-free and cell-free fluid that circulates through the ventricular system, so through these interconnected caverns within brain tissue. It moves out into the subarachnoid space that surrounds the brain. It's reabsorbed into the blood circulation through arachnoid villi, which sort of cross the boundary into dural venous sinuses. And the classical idea is that the CSF flow circuit is a one-way circuit. It moves from the site of production within the ventricles through these flow pathways and eventually out into the efflux pathways at the arachnoid villi. The function of the cerebral spinal fluid is well known and well appreciated. It creates buoyancy for the brain so that the brain doesn't have to support its own weight, and it provides a cushioning for the brain within the confines of the skull against injury. It has a role in volume balance within the skull. So as blood flow changes, changes in CSF volume can offset those changes in blood flow, so that pressure and overall volume within the cranium can remain constant. And it's long been appreciated that the CSF serves as a sink for metabolic wastes that are produced. So for wastes that are produced as a result of normal cellular metabolism, some of those wastes can be cleared across the blood-brain barrier easily, like carbon dioxide. Some can be locally degraded by cells within the CNS. But for wastes and solutes that can't cross the blood-brain barrier and can't be locally degraded, the way that they get out of the CNS is through some sort of exchange with the surrounding cerebral spinal fluid compartment. And the framework for this is termed the CSF sink hypothesis, which was articulated back in the 1960s. And it's framed around a couple of things. So one thing that is framed around is the anatomical observation that the CNS, so the brain and the spinal cord, lack lymphatic vessels, which means that the functions that lymphatic vessels subserve peripherally, which includes interstitial protein and fluid uptake, peripheral immune surveillance, those functions in the CNS have to be subserved in some other way. And so the notion is that the CSF serves as that terminal sink for solutes that are produced within the brain interstitium. And that exchange between the brain interstitial compartment for different solutes either internally with the fluid within the ventricles or externally with the fluid in the subarachnoid space. That process is slow, it's diffuse, and it's not necessarily physiologically regulated. So if you've heard any of my talks around the University of Washington in the last 3 years, you probably, or if you've even been paying attention really in the neuroscience space over the last 10 years, you know that major elements of this framework have come into revision over just the last decade. So chronologically, the second big finding that occurred was in 2015. Two different groups, one in Virginia and one in Finland, demonstrated that contrary to what everyone thought, there are in fact classical lymphatic vessels. They're not within the CNS, so they're not in brain tissue themselves, but they serve the CNS. So these meningeal vessels, they have all the markers of meningeal lymphatic vessels. They participate in the uptake of solutes from brain tissue and from the CNS. They're anatomically, they're embedded within the dura, typically associated with dural sinuses, although there are many of them also down in the skull base, just sort of sitting down there. So there are in fact lymphatic vessels, not in the CNS, but serving the CNS in CNS adjacent tissues that participate in the terminal clearance of solutes out of the cerebrospinal fluid compartments. That's an important topic, especially from the perspective of neuroimmunology, which is a very rapidly changing space, research space right now, but I'm not actually going to talk about it. What I will be talking about is this notion that the exchange between the brain interstitial compartment, which is the space between the brain cells, and the CSF that surrounds it, this notion that it's a slow process or a diffuse process or an unregulated process. And so this is sort of an experimental observation from a paper, the first paper that we published 10 years ago, that demonstrates that this is probably not true at all. So this is a pretty straightforward experiment. This is a study done initially in mice. So we're doing in vivo 2-photon microscopy, which is a fluorescence-based imaging through the skull of a mouse while it's awake. We've labeled the blood vasculature with a red tracer. So these are arteries here and veins at the brain surface. And then all of what you see down here, these are cerebral blood vessels down within the cortical mantle. We inject a fluorescent tracer into the CSF, so this is a 40 kilodalton dextran, and then we image its distribution over the brain surface over the next hour or so. And what you can appreciate immediately just by seeing this is that the CSF tracer, it's not moving diffusely over the surface of the brain, but rather it's following a very specific anatomical pathway. It's moving over the brain surface along the outsides of blood vessels, so along what are called perivascular spaces. And then it doesn't just sit on the outside of the brain, but rather begins to move into and through brain tissue along the outsides initially of penetrating blood vessels that dive down through the cortical mantle, eventually exchanging, and that's what you can see down in these lower tissues here. The video is a little out of sync now, but in this green here, this is a CSF tracer that is exchanged into and through the brain interstitial space. So it turns out that CSF, it's exchanged with the interstitial space. It isn't slow, rather it seems to be rather rapid. It isn't diffuse, rather it's anatomically organized. And whether it's physiologically regulated, I'll get to here in a second. This isn't something that's just happening microscopically up at the dorsal surface of the brain where we looked in that study in a mouse. It's something that's happening throughout the brain. So this is a very similar experiment. A couple of key differences being this is a rat and not a mouse. We're imaging by dynamic contrast enhanced MRI instead of fluorescence imaging. And instead of injecting a fluorescent tracer into the CSF, we've injected a gadolinium-based contrast agent, so gadoteridol, into the CSF that surrounds the brain. And then what I've done here is I've encoded the signal change from the contrast agent over the first hour in blue, the signal change in the second hour in green, the signal change in the third hour in red, so you can get a kind of a time course of what's happening. And what you can appreciate is that the CSF tracer, in this case a gadolinium contrast agent, moves into and through brain tissue along the axis of cerebral vessels. So it's actually using the cerebral arterial vasculature as a scaffold for organizing fluid and solute movement through brain tissue. So you can see the course right here of the anterior cerebral artery in the sagittal plane. You can see the choroidal arteries running here in the coronal plane. If we look over here, you can see the posterior cerebral arteries in the axial plane. And then you see in red the diffuse movement, the diffuse movement of contrast through brain tissue at later time points. So this demonstrates that the movement of CSF through brain tissue is anatomically organized along the structure of the vasculature. And this is something that's occurring across the entire macroscopic scale of the brain. Of course, if this was something that was only happening in mice, to be frank, you probably shouldn't care very much about it, but it turns out that this is happening in the human brain as well. So there's a group in Oslo, the University of Oslo, that has done a series of studies, essentially the same as this imaging study I just showed you in a rat, but in people. These are, what they're doing is they're injecting gadobutrol, another gadolinium-based contrast agent, intrathecally, so into the lumbar intrathecal space, and then serially imaging the participants over time to evaluate the distribution of that CSF contrast agent and how it interacts with brain tissue. And what you can appreciate, and this is a case series of 8 reference subjects, these are people being evaluated for things like CSF leaks, is that after intrathecal contrast injection, there's early filling of the basal cisterns with contrast agent. You see early, in the first few hours after injection, you see early movement of contrast into brain tissue. When participants come back the next day, so after going home overnight and then returning, there's a global and parenchymal enhancement of brain tissue. And so what this is the movement of contrast agent from the CSF compartment, which is now empty, largely empty of contrast, into and through brain tissue, demonstrating that this rapid exchange between the CSF and the brain interstitial compartment is happening not just in rodents, but is happening in the human brain as well. So to summarize, 10 years worth of work in our lab, but also many, many other labs that are now operating in this space, we now know that this perivascular, what is termed glymphatic, so this is, it's dependent on glial vessels and it subserves the lymphatic function, exchange, and meningeal lymphatic drainage. There's some things we know about it. We know that it's anatomically organized and rapid. We know that it's driven by arterial pulsations and vasomotor oscillations. We know that it's supported in an important way by astroglial water transport. We know that it seems to be a function of the sleeping rather than the waking brain. And we know that these two processes, perivascular exchange between the CSF and interstitial compartment and the clearance of solutes out of the CSF along these meningeal lymphatic drainage, these are physiologically coupled to each other. So these aren't distinct processes, but rather sequential steps in a linked process. Of course, you shouldn't take my word for this. You should never take a basic scientist's word for really anything. Make them show you the data. So if you want to see the data, follow this QR code. It will take you to a list of articles that show you that demonstrate these things that I'm telling you. Right? So you can either take my word for it if you want, or you can go check out more on the subject by following that QR code. So what I'm going to do next is I'm going to talk a little bit more about how it is that we know that this process is supported by astroglial water transport. We're going to talk a little bit more about how we know that it's more rapid in the sleeping brain, and then we're going to move on to some clinical and translational implications of this. So first, talking about the relationship between this process and sleep, I'm first going to talk a little bit about the relationship, our understood relationship between sleep disruption, and neurodegenerative processes. So going back to the case of Alzheimer's disease. So Alzheimer's disease is the most common cause of dementia worldwide. It's characterized histopathologically by a couple of features that I mentioned before. So extracellular amyloid plaques, intracellular neurofibrillar tangles. There's a long understood association between sleep disruption and Alzheimer's disease and actually dementias of all kinds. Right. So actually, there was a paper that I cite to students when I'm lecturing in the neuroscience PhD program on the relationship between sleep and Alzheimer's disease. There's actually a paper from Elaine Peskind here at the Puget Sound, VA Puget Sound, back from 1980. I think it was 81 or 82 when I was 2 years old. She actually published a paper reporting that people with dementia of the Alzheimer's type had really profound sleep disruptions. And the assumption really for a long time has been that clinical association between profound sleep disruption and dementia and Alzheimer's disease is the result of the degeneration of sleep-regulating brain centers. So the idea is with progressive neurodegeneration, the parts of the brain that regulate the sleep-wake cycle become dysfunctional, and hence that clinical association. But a couple of threads of evidence that have emerged over the last 5 to 10 years have suggested that that relationship may be more complicated and more bidirectional than we appreciated. So there's two things that I want to draw your attention to. One is, this is one study, but it represents actually a family of studies from David Holtzman's group at Washington University in St. Louis. And what this group has demonstrated is that there is a diurnal fluctuation in proteins and peptides, including amyloid beta, including tau, and including alpha-synuclein, where their levels in the CNS and surrounding the CNS oscillate with the sleep-wake cycle. So this is actually a really interesting experiment where they're conducting continuous CSF sampling from participants through the course of a day. And what they observe is that lumbar CSF levels of amyloid beta increase during the waking day and then decline overnight, increasing the next day. This diurnal fluctuation in amyloid beta shown here in this paper in 2009, and in more recent studies, they demonstrate the same thing with tau and alpha-synuclein, suggests that there's an important hour-to-hour, day-to-day regulation of the levels of those proteins, which are the proteins that aggregate in neurodegenerative disease, that links them to the sleep-wake cycle. Another series of studies have demonstrated that even in people before in the absence of frank cognitive impairment, so in cognitively normal individuals, there's a connection between sleep behavior, whether that's sleep duration or sleep quality, and the development of Alzheimer's related pathology. So this is one such study from Adam Spira at Hopkins, where they evaluated self-reported sleep duration in cognitively intact people who underwent assessment of brain amyloid burden using positron emission tomography. And the simple finding from the study was that in people with a self-reported sleep duration of less than 6 hours, they exhibited a higher amyloid burden than those who exhibited self-reported sleep durations more than 7 hours. And they observed a similar effect with people who reported poor sleep quality. So there's a sort of a building corpus of clinical and observational research that suggests that there is actually a relationship between sleep disruption, including sleep disruption in midlife, and both the risk of dementia and the risk of Alzheimer's disease, and the development of Alzheimer's related pathology, all in human populations. So there's several of these studies that sort of form this idea. I don't have time to go into it right now, but if that's something that's of interest to you, this is a different QR code. You can scan this code and it will take you to a family of articles that get at this, both from the epidemiological side, from the neuroimaging side, and from a couple of different perspectives. So if it's true then that there's a relationship, that there may be a relationship where sleep disruption may be providing some sort of vulnerability to the development of Alzheimer's related pathology, the question arises about whether this process of perivascular exchange, this lymphatic clearance, whether this is regulated itself by the sleep-wake cycle. So this is going back in time a little bit. This is a paper that was published back in 2013 in Science, essentially asking that question in mice. So this is a pair of experiments in the same animal. In the first case, this is the same kind of imaging study that I showed you before. So we have a mouse, we're imaging down into the cortical mantle with 2-photon microscopy. This is a field of view down within brain tissue itself. This is an animal that's awake at the time of imaging. This is the same animal later on in the experiment when it's been anesthetized with a cocktail of ketamine and xylazine. And the observation is that in the waking state, it looks like maybe my movie isn't playing. In fact, it is. It's just that there's very little movement of CSF into brain tissue in waking animals. Whereas when that same animal is anesthetized, the situation completely changes. We can quantify that here on the right. So you can see in the waking state, there's very little movement of CSF tracer through brain tissue. In the anesthetized state, it's much more rapid. Of course, sedation and anesthesia are not the same as sleep. So when animals are experimentally sort of driven towards sleep using a sleep deprivation recovery sleep paradigm, we observe the same sort of thing where animals that are sleeping on the scope, they exhibit higher rates of CSF tracer movement through brain tissue than animals that are waking. Of course, this isn't just the movement of CSF fluid and solutes through brain tissue, it's also the clearance of solutes out of brain tissue. So it's how things are getting out, which is when we start talking about the subject of waste clearance, that's really what we want to be thinking about. So this is an experiment where we've injected two different radio tracers at the same time into animals. So one is an inert kind of generic solute. So this is C14 labeled inulin, which is about a 5 kilodalton polysaccharide. We also inject iodinated amyloid beta 40 into brain tissue. We collect the brain after certain time points and measure how much of the radioactivity is left in it. From that, we can derive clearance curves and rate constants that give us a snapshot of how fast interstitial solute clearance is happening. And what you can appreciate is that both for the generic solute inulin and for amyloid beta, there's about a 2-fold increase in the rate of clearance in either animals that are sleeping or anesthetized with this particular cocktail of ketamine and xylazine compared to the waking state. So what this demonstrates is that, at least in mice, there's both more rapid CSF movement through brain tissue and more rapid clearance of interstitial solutes out of brain tissue in the sleeping compared to the waking brain. As I mentioned before, you shouldn't care about any of this if this is only true in mice, or if maybe, I don't know, maybe you're a mouse veterinarian or something like that, or you really care about mice. But we're in the business, though, of trying to target and improve human health. So if this isn't something that's happening in the human brain, I think interest in it should be diminished. And so that's why I think this paper here that was published by that same group in Oslo I mentioned a couple of minutes ago is so important. So this was the first confirmation in human participants of sleep-active glymphatic clearance. So the design here is pretty interesting. So as I mentioned before, what these investigators are doing is they're doing intrathecal gadobuterol injection and then serial MRI imaging afterwards. So they took their participants, they injected them with IT gadobuterol, they waited somewhere around 6 hours or so, and they see about equal uptake in the gray matter of contrast agent into brain tissue. And then they take the two groups, and half the participants are allowed to sleep normally overnight, and the other half of the participants are required to stay awake, or they're kept awake all night. So this is a total sleep deprivation paradigm. And what they observe is that the clearance of this contrast agent out of brain tissue in the overnight period is slowed in the sleep deprivation state. In fact, when participants are allowed to sleep on the second night, right? So they get to have normal sleep. Both groups get normal sleep on the second night. That difference persists, suggesting that the deficit in clearance that results from a single night of sleep deprivation takes more than one night for it to resolve. When the participants returned 4 weeks later for assessment, no one had any contrast left in their brain. So sometime between 48 hours and 4 weeks later, things had normalized. But what this demonstrates is that this sleep-active CSF interstitial fluid exchange is happening in the human brain as well as in the rodent brain. So if that's true, if this sleep active process is contributing to the clearance of solutes like amyloid beta, like tau, and like synuclein, then the question arises whether its impairment may contribute to the vulnerability of the aging or the injured brain to Alzheimer's related neuropathology. So for the next little bit, I'm going to talk about some things that we've learned about changes in glymphatic function in the aging and the injured brain. So this experiment, this is something we reported in Annals Neurology in 2014. This asks the very simple question of, is glymphatic impairment impaired in the setting of aging? Not in the setting of any other comorbid condition other than just aging. And it's a little bit, it's kind of debatable whether that's a pathological condition, right? It's just a sort of a physiological state, right? And so the experiment's pretty straightforward. It's similar to what I've shown you before. We're injecting, in this case, 2 different tracers, a red and a green one of different sizes into the CSF. We let the animals wait for 30 minutes, and then we sacrifice the animals and evaluate CSF movement through brain tissue. And what you can immediately observe is that in healthy young adult mice, there's rapid and extensive exchange through brain tissue. In the aged animals, so in 18-month-old mice, and those are pretty old, but not the oldest mice you can get. So in pretty old mice, we see that CSF movement through brain tissue is slowed. When we measure the clearance of solutes out of brain tissue, we see the same process. So here we're injecting in radiolabeled amyloid beta into the striatum. We measure how much of it is cleared in the first hour. So in the first hour of healthy young animals, we see about 70% of the amyloid is cleared in 1 hour. We see in 12-month-old, which is a middle-aged mouse, in 18-month-old, which is an old mouse, we see a stepwise slowing in the clearance of interstitial amyloid beta out of brain tissue that parallels the slowing of CSF movement through brain tissue. So what this tells us is that in just the old brain, this process of fluid exchange through brain tissue and the clearance of amyloid beta does seem to be slowed. I'm not going to go into it. I don't have anywhere near the time to do so, but other studies from other groups and from our group have demonstrated since 2014, so over the last 8 years, that similar impairment of glymphatic function is observed in the setting of, in animal studies, experimental traumatic brain injury, ischemic injury, subarachnoid hemorrhage, cortical spreading depolarization, which is believed to be the neurophysiological basis for migraine aura, animal models of Alzheimer's and Parkinson's disease, animal models of chronic hypertension and diabetes. These are important risk factors, of course, for dementing disorders. In the limited number of studies that have evaluated this in human populations, we see evidence of glymphatic impairment in people with normal pressure hydrocephalus, evidence of cerebral small vessel disease, and people with Parkinson's, people with Alzheimer's disease. And of course, please don't take my word for it. If this is something you're interested in, you want to find out if I'm just making stuff up, or if this is really something that's holding water, go check out this QR code that will take you to another list of articles that demonstrate these findings both in rodent and in human populations. So this is a rapidly expanding field. But the takeaway here is it appears that impairment of this perivascular exchange seems to actually be a common feature of both the aging and the injured brain. So if this is a feature of the aging and the injured brain, what is it that's changing in the aging brain that underlies that impairment? What's causing it? And the caveat that I'll provide here is that I'm going to talk a lot about this particular molecular entity, so aquaporin 4, which is one of my lab's favorite molecules. Every lab has a favorite molecule, and every lab thinks that it is the most important thing in all human disease, and we think that too. The caveat is we think a lot of things are changing in the setting of aging. There are all sorts of cerebrovascular changes, right? There's all sorts of changes in like the extracellular matrix. There's all kinds of changes happening in aging. This is one that we've spent a lot of time studying. We think that this is an important one. We do not think it is the only one, but it is the only one I'm going to be talking about for the rest of this talk. So I'm going to talk about aquaporin 4. Aquaporin 4 is a protein. It's a transmembrane protein that forms a pore that allows water, so it's a water selective channel, to diffuse down its concentration gradient, facilitating water transport across biological membranes. There are aquaporins really across the animal and the plant and the other kingdoms, right? In the brain, there's mainly 2 aquaporins are expressed, aquaporin 1 and aquaporin 4. Aquaporin 1 is expressed in the choroid plexus, where it's involved in CSF secretion, which is the subject of a different talk. Aquaporin 4, here it's labeled in purple in the rat brain. You might think based on the labeling that it's a vascular protein, but it's not. Rather, it's an astrocyte-specific protein. So astrocytes are the kind of support cells of the CNS. It's astrocyte-specific, but it localizes to the part of the astrocyte that lines the blood vasculature. And so what we think aquaporin 4 is doing is it's organizing the movement of water through brain tissue along the axis of the vasculature. And by creating that kind of scaffolding in that organization, it makes that process more efficient. So in our first paper back in 2012, we evaluated the role of aquaporin 4 in this perivascular exchange. And one of the most straightforward ways to do this is to evaluate this perivascular exchange in animals where the gene encoding aquaporin 4 has been deleted, right? So this is the same type of study I've shown you before. You should at this point be kind of used to seeing it. Here we're injecting a 45 kilodalton fluorescent protein into the CSF. We measure its movement through brain tissue in wild type animals. So in the first 30 minutes, there's pretty extensive movement of the tracer through brain tissue. In animals that lack the aquaporin-4 gene and don't express the aquaporin-4 protein, we see that the movement of CSF tracer through brain tissue is markedly suppressed. We see at the same time a suppression in the clearance of solutes out of brain tissue. So here we're injecting two different tracers, one an inert generic solute, in this case tritiated mannitol, and the other radiolabeled amyloid beta, into the brain of wild type and aquaporin-4 null mice. We then measure the rate of their clearance out. We observe that in wild type animals, so animals with aquaporin-4, there's a certain rate of clearance out of brain tissue. So in the first hour after injection, about 70% of the amyloid is cleared out of the wild type brain. In the mice that lack aquaporin-4, we see that there's a marked suppression in the clearance, both of a generic small solute like mannitol, but also of amyloid beta, suggesting that this process of CSF movement into and through brain tissue is dependent on the astroglial water channel aquaporin-4. So then if this process is dependent on aquaporin-4, you might suspect that maybe it's the loss of aquaporin-4 that contributes to the clearance, that contributes to the inhibition of this exchange in the aging or in the injured brain. We can test that. We can just measure aquaporin-4 expression in young and old animals. In this case, we measured it by Western blot, which is a way to measure protein levels. We compare between 2 to 3 month old and 18-month-old animals. And actually what we see is that it isn't that it declines, but rather aquaporin-4 expression in the aged animal goes up. It's markedly upregulated. But if you look at it using immunofluorescence, look to see where it is in brain tissue, what you appreciate is that while there's higher levels of it, and here aquaporin-4 is labeled in purple, in aged animals here on the bottom and young animals on the top. So there's higher levels in the aged animals, but its distribution has changed. So in a young animal, if we look at the level of a single astrocyte, most of the aquaporin-4 is localized mainly to the part of the astrocyte that faces the blood vessels. But in the aged animal, the aquaporin-4, it loses the fidelity of that localization. So it ends up being spread over all of the fine processes of the astrocyte. So this loss of perivascular aquaporin-4 localization, this is the phenotype that we see in aging, and it's actually the phenotype that we see in the setting of injury. I'm not going to show you that right now. So the question is this loss of perivascular aquaporin-4 localization, is this what's impairing the glymphatic function? Is this something that could contribute to the development of Alzheimer's related pathology? So it turns out that we know a little bit about why aquaporin-4 has this perivascular localization, and there are some useful tools in evaluating the role that that localization has. So we know that aquaporin-4 maintains that perivascular localization through its coupling to a macromolecular complex that binds it to the extracellular matrix of the blood vasculature. That's out here. So aquaporin-4 binds to alpha-syntrophin, dystrobrevin, dystrophin, and dystroglycan. So there's this series of proteins here that anchors it to the perivascular input. If you knock out any one of the genes that encode these proteins, this is the phenotype that you get. So you get normal levels of aquaporin-4 expression, but instead of it being mainly perivascular, it ends up spreading out over all the processes of the astrocyte. So this is at least a rough phenocopy of what we see in the aging and the injured brain. And so we can use this animal to test whether this kind of loss of perivascular localization, whether it has any consequences. So this is a pair of studies that we've published first in 2018 and more recently this last year, evaluating this question. So first, we can measure this perivascular exchange in animals that lack perivascular localization. So this is a similar type of a study to what I showed you before. So this is contrast-enhanced MRI. We're injecting a gadolinium-based contrast agent into the CSF around the mouse's brain. We then image over time the movement of the contrast through brain tissue. So between 30, 60, and 90 minutes, you see increasing enhancement of brain tissue as that CSF tracer moves through the brain tissue. In the alpha-syntrophin knockout mice, we see that enhancement is slow, meaning there's less movement of fluid and solutes through brain tissue. This is, we've now shown this, this next finding we've now shown twice in two different transgenic lines. So we wanted to ask the question is, does loss of this localization, can it promote Alzheimer's related pathology? And so what we've done is we've crossed this transgenic line that doesn't have alpha-syntrophin, so these lack perivascular localization, we've crossed it into a couple of different transgenic mouse lines that naturally make amyloid plaques. And when we do that, what we observe is a pretty marked increase in amyloid plaque burden when animals don't have perivascular localization of aquaporin 4. So they have aquaporin 4, it's just not where it's supposed to be. So what this demonstrates is that loss of perivascular localization of aquaporin 4, it can impair lymphatic function, and it can promote the development of Alzheimer's related pathology. What it doesn't tell us is that this is what's happening in the setting of human Alzheimer's disease. We don't know that this is actually the series of events that's contributing to the development of Alzheimer's related pathology and the eventual development of clinical AD. So what I'm going to finish with here are a pair of studies where we're trying to get at that question, trying to look at whether there's a role for these changes in aquaporin 4 in the development of human pathology and human disease. So one is a histopathological study, and one is a genetic study. And these give us two different views of the same process. So this is a histopathological study that we did in collaboration with the Oregon Brain Bank down at OHSU. So this is something similar to what we showed you before, looking at aquaporin 4 localization in the rodent brain, except here we're looking in the frontal cortex of human postmortem samples. Now we have samples from cognitively normal participants, participants with histopathologically confirmed Alzheimer's disease, and participants that died with mild cognitive impairment, so sort of an intermediate cognitive phenotype. If we measure perivascular localization in the gray matter of the frontal cortex, what we observe is that in participants with Alzheimer's disease, there's a loss of perivascular localization. So it's less perivascular in people with AD compared to cognitively normal controls. And MCI has sort of an intermediate phenotype, which is maybe what we would expect. The interesting thing is we observe that this perivascular localization, though it's a histological feature, it correlates really well with a couple of with features that are related to disease progression. So if we look in the earlier stages of the disease, so in people that are cognitively normal or people with MCI, we observe that loss of perivascular localization, which is movement on the x-axis to the left here, is associated with poor performance on MMSE, Mini Mental State Examination, and the clinical dementia rating sum of boxes. So these are two commonly used measures of cognitive and functional decline in dementia populations. So this suggests that changes in perivascular localization are at least associated with features of cognitive impairment in people at early stages of disease. When we measure local changes in Alzheimer's pathology, so the CIRAD score measures neuritic plaque burden, so those are amyloid plaques. Phospho-tau density is measuring how much tau pathology there is in the cortex. We see the same sort of the same correlation, where declining perivascular localization is associated with higher amyloid and higher tau burden, at least in participants in the earlier stages of the disease, so in cognitively normal participants and in those with MCI. These correlations go away in people that die with Alzheimer's disease, so in the end stage of the condition. Of course, this kind of a study doesn't tell us whether the pathology, the Alzheimer's pathology, is causing the changes in aquaporin 4, or whether the changes in aquaporin 4 are causing the pathology. This is an associative study, so it can't get us at causation, right? So one of the approaches that we've taken to trying to get a sense of a potential causal role for changes in this perivascular biology and in the development of Alzheimer's disease is to look at gene association study, is to look at genetic studies evaluating the relationship between naturally occurring variants in the genes surrounding this biology and vulnerability to different pieces or phenotypic features of people with Alzheimer's disease. So this is a collaborative study that we did out of the Alzheimer's Disease Neuroimaging Initiative cohort. So this includes participants that were cognitively normal, participants that had MCI, and participants with Alzheimer's disease. We evaluated a series of naturally occurring variants in the human genes encoding aquaporin 4, dystrobrevin, dystoglycan, and alpha-syntrophin, which are the genes that define perivascular aquaporin 4 localization. We evaluated the relationship between having these single nucleotide polymorphisms and these different things. So cognitive status, so whether the person is intact or impaired, CSF-AD biomarkers, so CSF A-beta 42, total tau, phospho-tau, longitudinal cognitive decline, so longitudinal decline in memory or executive function, longitudinal measures of atrophy, whether that's hippocampal atrophy or cortical atrophy. And in our models, we included as covariates age, sex, ApoE status, education, and for the longitudinal values measures, we included the baseline value as a covariate. I'm not going to go through the really dense tables because that would be boring, and frankly, I don't have enough time. I'm going to cut to the punchline. The punchline is, of the 11 aquaporin 4 SNPs that we evaluated, 3 of them were associated with different, either higher or lower levels of CSF total tau, CSF phospho-tau, measures of longitudinal measures of cortical atrophy and hippocampal atrophy. Of the 40 tag SNPs in the dystrobrevin gene, 15 were associated with either CSF A-beta levels, CSF total tau levels, longitudinal memory decline, longitudinal cortical or hippocampal atrophy. Of the 4 SNPs in the dystoglycan gene, 2 of them were associated with hippocampal atrophy. And of the 1 SNP that was present in the syntrophin gene in the SNP array that was used, it was associated with variation in CSF A-beta levels. This doesn't tell us that necessarily these genes and their products have a causal role in the development of Alzheimer's disease, but they provide corroborating data that suggests that something's going on here at the perivascular infant that seems to be conferring either vulnerability to or protection from these different features of Alzheimer's disease, whether that's a development of pathology or whether that's progression that includes cognitive decline and eventual degeneration of brain tissue. There are several studies that have corroborated these findings. We haven't published these findings yet, but other studies have published similar findings. If you're interested in those associations, you can check them out. Go to this QR code and it'll take you to them. So a quick summary, and then I'll do an implication next. Quick summary is, if you haven't heard of it yet, if this is new to you, the glymphatic pathway is a brain-wide perivascular network that supports the exchange between the CSF outside the brain and the interstitial fluid within the brain. And it appears that exchange seems to be a feature primarily of the sleeping brain. In data from animal models and increasingly from human participants, we see that it is impaired in the setting of aging and in the setting of conditions that we know to be risk factors for the development of neurodegenerative conditions, including sleep disruption, cerebrovascular disease, traumatic brain injury, and others. What we think, what our lab thinks and what the others in the field now think, is that impairment of glymphatic function seems to be a general feature of the aging and injured brain, and that loss of this clearance may be one of the features that makes the aging and the injured brain vulnerable to the development of the pathology associated not just with Alzheimer's disease, but other neurodegenerative diseases as well. Why does that matter? Okay, so this is with my last sort of 2 minutes of talking here. I want to get at this point. Why do we care about this? We care about this because we think that this is a biological process that is upstream of the development of Alzheimer's related pathology. So everyone knows about the serial failure of amyloid targeting therapeutics in the Alzheimer's space. It's a big news story. Everyone knows it, and it's sort of creating a crisis of faith almost within the Alzheimer's field. The thinking, one school of thought in the field is that what's happening is we're not targeting the development of amyloid or tau pathology early enough. And then if we could only get far enough ahead before unstoppable processes become unstoppable, then we could actually bend the curve a little bit. The interesting thing about this biology is when we think about the clinical progression of Alzheimer's disease, or the progression of Alzheimer's disease, the last feature of which is cognitive impairment and clinical progression, tau pathology, which is this blue curve, and amyloid, the development of amyloid pathology, which is this red curve, these are happening probably decades before the onset of clinical symptoms, right? So if we could identify something that's to the left even of these processes, then that would give us an opportunity to perhaps prevent the development of Alzheimer's related pathology and eventually the development of the disease. So our thinking is that lymphatic impairment, if it contributes to amyloid pathology and contributes to tau and synuclein pathology, then it sits to the left here. And so if we could detect lymphatic impairment in people, particularly in midlife, then it may offer us a window into who is it that is vulnerable to the development of Alzheimer's related pathology, maybe even before that pathology has begun to take root, or certainly within the earliest stages of the development of that pathology. More interesting still is in these vulnerable individuals, if it were possible to preserve lymphatic function and preserve this clearance process, it may actually enable primary or secondary prevention in this disease state, right? So sort of like targeting hypertension in the prevention of heart disease, it may be possible to target lymphatic impairment, the impairment of clearance, in the prevention of amyloid and tau pathology, and thus the prevention of Alzheimer's related dementia. So that's it. I'd like to acknowledge, this is my lab here on the left. These are my really incredible collaborators who are trying to do lymphatic imaging in the human brain. My incredible collaborators at the VA Puget Sound, who really were a big reason why I came here in the first place to the University of Washington. So Gail Lee, Elaine Peskin, Murray Raskin, and Abby Schindler. These are incredible people to work with. My lab we are constantly expanding. We're always looking for new and interesting people who want to study this biology in new clinical populations. So if this is of interest to you, this last QR code will take you to my lab's website and will tell you how to get in touch with us and how we talk about maybe working together on this in the future. So with that, I'm glad to take questions, if there are any.
Well, thank you, Dr. Iliff. A lot of new information, and thinking through specifically in clinical scenarios and trying to relate these discoveries and findings, really helping us all think about this in new ways, frankly. And I think there are a couple of questions that have come in, and I have some as well. One that came in is about, it looks like the images that you showed maybe showed the lymphatic system more active in the cortex rather than deep structures. Is that likely what's occurring, or is that based on measurements? Yeah.
I think that's really good. So when so I think there's an important caveat in that the way that we measure lymphatic function is by putting in a contrast agent at a point source and then visualizing its distribution elsewhere. And when we think about the organization of that movement, it starts at the proximal vasculature and it follows the vasculature out. And so when you're talking about deep brain structures, those are kind of the bitter end of many of them are in the bitter end of the vasculature, right? And so one possibility is that it's just as fast in those regions. It's just that there's so little tracer left to get there that we can't detect it, right? We are currently using non-contrast methods that capture lymphatic function. And those, I don't know, if I had to guess, I think that there's maybe more activity in the cortex. We do see that that cortical changes, so for example, in aquaporin 4, those seem to be the things that are changing with aging much more than the deeper structures. So when we look in striatum and we look in sort of diencephalon, we don't see such pronounced differences as we see in the cortex. So it may be that there's an important role for, especially when you think about the gyrencephalic human cortex, there may be actually a really important role for access to fluid in these processes that might be play out a little bit differently in older regions of the brain.
Thank you. I did get an FYI message that one participant was seeing that the chat is disabled. I was entering things in the chat. If you're not able to write a question in the chat, you could write it in the Q&A. I am seeing questions that are coming in there. I couldn't make an adjustment, but just use the Q&A. That seems to be working fine. Here's some related questions came in, and I was wondering about sleep duration. You showed an experiment based on duration of sleep. And is it just duration, or is it architecture of sleep? Is it sleep efficiency? Is there a stage that's more associated with exchange? What
So,
about disruptions related to mood disorders, for example? Yeah,
yeah.
a range of comments, perhaps.
So let's talk about sleep first, mood disorder second. Sleep first. So I would say that we are just starting to pull that apart. The data so far has all been in rodents, has almost all been in rodents. And rodent sleep structure is really different than human sleep structure. One of the key differences is the consolidation of sleep stages into really long swaths of time that you see in humans normally. Then in rodents, it's actually really fragmented. So there is people think that it's slow wave sleep that's primarily associated with. I actually think the data for that is really weak. I think it makes a plausible sense, but I don't think anyone's actually ever shown that in humans. And I think that we're sitting at a time when defining the relationship between sleep microstructure and this process, we're now ready to answer that question, but I don't think anyone has ever answered it. I think there's a case to be made electrophysiologically and physiologically why slow wave sleep would be involved. I think there's a case to be made for why REM sleep might be involved, but no one has demonstrated it. There's an interesting idea, I actually just read it this last week in a recent paper, having to do with inertia. So when you think about fluid movements, when if you stir a bucket of water, when you first start stirring, all you get is turbulence, but it takes a while and eventually you get this big coherent whirlpool. And so it's possible that the coherence of the human sleep state, right, which is sort of, N3 stages of slow wave sleep are pretty protracted over long periods of time. It's possible that actually might be important for especially given the scale of the human brain, to develop fluid inertia like over the length scales that you have in the human brain, whereas in a mouse brain, the mouse brains are, they're this big, and maybe it's not so necessary, right? And so maybe that's evolutionarily why their sleep is sort of permitted to be so fragmented. So I think the jury's really still out on there. Going to the question of mood disorders. So everything I've talked about up to this point has all been through the lens of neurodegenerative disease. We've just talked about waste clearance, whatever waste is. Waste, the only things that have been demonstrated to be cleared by this pathway include amyloid beta tau, alpha-synuclein, and lactate. No one has evaluated anything else. So because we came to this biology through the lens of Alzheimer's disease, that's the only thing we've thought about. But the same process that we're talking about is probably involved in cytokine dynamics and clearance. It's probably dictating the dynamics of neuromodulators, which are released not synaptically, but released by volume transmission into this extracellular space. So things like serotonin and norepinephrine, right, and acetylcholine. These are things that are very active in mood and implicated in mood disorders. So the fact that many of those disorders are connected with sleep disruption, I think that that's an untapped sort of unexplored continent right now, where we now have the methods to measure glymphatic function in people. It's just that no one from the psychiatry space who has an interest in mood disorders has started to look at that question. It's all been people from the cerebrovascular side and from the neurodegenerative side.
It's very, we were talking before the presentation started about new ideas coming up during grand rounds, and maybe that inspired someone.
I really hope so. I really hope
you'll hear it from someone next week. Let's hope. I mean, I guess just a follow-up comment. I think there's been observational studies of associations of depression and incident dementia later on, and one can think about ideas related to that.
Well, so the interesting, and this is maybe a non sequitur. We've published the human aquaportin 4 finding. We've now published it twice in two different case series. So, but the immediate predecessor to that, when we were trying to evaluate, what do we know about aquaportin localization in the human brain? The best case series was actually from a case series focusing on major depression. And the observation was that age at death was inversely related to perivascular localization. So the younger they were when they died, the worse the perivascular localization was, which to me didn't make a lot of sense, because I was thinking about it very simplistically, where, mice, we know why mice die. They die because we kill them, right? Which is like a sad reality. But in the setting of major depression, one of the most common, at least in that case here, is one of the most common causes of death was suicide. And so the interesting implication there might be that early death in that setting was associated with changes in perivascular localization of aquaporin 4. Now that wasn't evaluated really in that way. It's just sort of an interesting, maybe related finding that might be a little bit of a teaser or a little bit of a hint that there's something worth looking at there.
Here we are just at 1 o'clock. Perhaps you can just give a quick comment. You got to it a little bit with this idea about stirring water in a bucket. Maybe it's related. Why might the brain cleansing, so to speak, why might not that occur during the daytime? Why is it linked to sleep?
So I don't think anyone has a really good, I think evolutionarily, it probably has some. So one of the findings from that 2013 paper was that the brain extracellular space actually changes in dimensions between waking and sleep. And during waking, it's smaller, and during sleep, it's like 50% bigger, right? It's a big difference in extracellular volume. And so it's likely that change in volume, that has really important consequences for ion transport, for neurotransmitter release, uptake, and the kinetics of those things. So I suspect, if I had to guess, there's something about that tight space which is important for the asynchronous activity that is characteristic of waking function. But with a smaller space, diffusion is markedly slowed. So the size of the space has a huge influence on diffusion, right? And so it's possible that the tight space that makes it possible for high fidelity transmission also keeps clearance from happening efficiently. And so it has segregated, slow wave sleep is really interesting, right? Where you have these oscillating periods of synchronized on and offness, right, that's happening. And it could be that segregation allows for some sort of a clearance process that isn't possible in the asynchronous activity that you see in the waking brain.
Well, thank you for your comments here in this series of questions. And Dr. Riley, thank you for presenting today at Grand Rounds. We're just a minute or two past 1 o'clock, and we need to end due to time. But this is it's very interesting, and I think for me, it's raised a lot of questions and interesting ideas. A lot of this is novel for me, and I hope that's the case for others as well. And I very much appreciate you presenting today. Thank you.
Good, thank you.
All right, bye.